Glycolysis and gluconeogenesis are coordinated, interrelated processes that ensure the energetic needs of all cells are met. Without these processes, cells would be severely energy deficient and would not live for any significant amount of time, and therefore are extremely important biological processes. Glycolysis is a process used by cells to obtain energy from food. The catabolic process of obtaining energy from food has 3 steps. The first step is when polymeric molecules are split into simple monomeric subunits. Proteins are split into amino acids, and fats into fatty acids and glycerol. And polysaccharides are split into simple sugars such as glucose. The second step is called glycolysis, and this is what we're going to focus on today. Glycolysis is how cells harvest useful energy from the chemical bond energy locked in sugars. Using the products from step 1 of catabolism, glycolysis changes glucose into pyruvate with ATP and NADH as energy byproducts. This process occurs in the cytosol of the cell and utilizes enzymes to break the high-energy covalent bonds of the sugar throughout 10 steps before reaching the final product. While there are 10 steps in glycolysis, 3 of these steps are so energetically favorable that they are irreversible. The first of these irreversible steps is step 1 of glycolysis, in which an enzyme hexokinase catalyzes the reaction of glucose with ATP to produce glucose 6-phosphate and an additional byproduct of ATP and H+. The second irreversible step is step 3, where fructose 6-phosphate and ATP are catalyzed by an enzyme called phosphofructokinase to produce fructose 1,6-biphosphate along with ADP and H+. The final step of glycolysis is also irreversible. This step occurs when pyruvate kinase catalyzes the reaction between phosphoenol pyruvate, ADP, and H+, to form the final product of pyruvate and ATP. Overall, the net result of glycolysis is the production of 2 pyruvate molecules along with 4 ATP and 2 NADH, which is energetically favorable considering that it only requires the input of 2 ATP to produce the end products.
Gluconeogenesis is a set of enzyme-catalyzed reactions by which glucose is synthesized from small organic molecules such as pyruvate, lactate, or amino acids. Basically, gluconeogenesis increases a cell's available glucose levels by synthesizing glucose from mainly pyruvate. It can be thought of as the reversal of glycolysis, as it builds glucose from pyruvate, where glycolysis breaks down glucose into pyruvate. It even uses mainly the same enzymes, it just runs them in reverse. Gluconeogenesis is able to get around steps 1, 3, and 10 by using a special set of enzymes that catalyze bypass reactions. For example, in step 3 of glycolysis, the enzyme phosphofructokinase, PFK, phosphorylates fructose 6-phosphate into fructose 1,6-biphosphate. In gluconeogenesis, the enzyme fructose 1,6-biphosphatase removes a phosphate from fructose 1,6-biphosphate, generating fructose 6-phosphate. Some bypass reactions required for gluconeogenesis are energetically costly. An example of this is the reversal of step 10 consumes 1 molecule of ATP and 1 molecule of GTP. Altogether, it costs 4 molecules of ATP and 2 molecules of GTP to produce a single molecule of glucose through gluconeogenesis. Because of this, gluconeogenesis processes must be very closely monitored by the cell. Glycolysis and gluconeogenesis are coordinated, interrelated processes that ensure the energetic needs of all cells are met. Without these processes, cells would be severely energy deficient and could not live for any significant amount of time, and therefore are extremely important biological processes.